Amino acids we talked about in our last video are organized into peptides. And peptides are generated by ribozymes, which are RNA molecules and that are able to catalyze specific biochemical reactions. For example, the condensation reaction forms peptide bonds, as you see here. And as a small reminder, peptide bonds are the bonds that are formed between the carboxyl group of one amino acid and the amide group of a second amino acid. And peptide chains are always formed from N to C, because the amide group of the first amino acid will always be the first one, and the carboxyl group will be the last open. So from N to C. And this is why proteins have an N terminal, which is the amide group, and the C terminal. And trick to remind you that proteins are built from N to C is, for example, an amino acid is NCC. And if you think about CNN and if you just switch the letters, it will be CNN, NCC. CNN backwards will be NCC. Peptide bonds are really stable because of the covalent bond between the carboxyl group and the amide group and the ressonance effect, which will form a partial double bond between these two atoms. So what is the ressonance effect? Resonance is a way of describing bonding in certain molecules or ions by the combination of several contributing structures or forms, also variously known as resonance structures or canonical structures, into a resonance hybrid or a hybrid structure in a valence bond theory. It has a particular value for describing dealkalized electrons with certain molecules or polatomic ions, where bonding cannot be expressed by one single Lewis structure. In this particular case, the oxygen of the carbonyl is a lot more electronegative, and this will cause a pull of the electrons to the oxygen, which will leave the carbon even more electropositive, which will make him pull the lone pair of electrons from the nitrogen, forming a partial double bond between the carboxyl group and the amide group. And this is why peptide bonds are so stable. But of course, in nature, the electrons will have a hybrid structure, as we've referred before. In the resonance effect causes a hybrid structure. Formation will make nitrogen hybridization something between sp3, which has a 109 angle and has a tetrahedral conformation, and sp2, which is planar and has an angle of 120 degrees. Since peptide bonds are so stable, in order to break this bond, you need either highly acidic or highly basic conditions. Basic conditions, the hydroxyl group will attack the carboxyl group, and the electrons will be kicked up and down, and you'll be left with a very negative, very unhappy nitrogen, because the sodium here is just a spectator. So this makes the reaction very slow and not a very good option. In acidic conditions, we will have the water attacking the carbonyl group, leaving one hydrogen to protonate the amide group, and this will make for a much more happy nitrogen. However, this is still not the best reaction to occur, and in nature, proteins are employed to degrade peptide bonds, and these proteins are called proteases. And when you see a word in biology ending in ases, you know it's an enzyme. And proteases are the enzymes that hydrolyze peptide bonds, whether it being endoproteases or exoproteases. But I will talk more about proteases in the next video. See you there.